Okie dokie. So welcome to part 2 of the chapter 3 business here. And this is starting on the proteins, and this is the table right in your book here, and it's talking about the different functions. So you go ahead and you look at the functions here. All right, oops, sorry, I kind of covered over that. Let's undo that. Can I undo that? Nope. And you have your different functions, and your functions, I'm going to play around with this here. Let's do my eraser. There we go. I erased it. Okay, so the functions are the same ones listed in your text, and this gives you examples of specific proteins that would be involved in these functions. So in defense, this is your immune system proteins, essentially, your immunoglobulins, your antibodies, and these are extraordinarily important, obviously, to your body, so proteins function in defense. I skipped enzyme catalysis. We already did that a little bit in our lab. Okay, different ACEs, okay, oftentimes they end in ASE, and these are enzymes that either bring things together or chop things apart, okay. Transport, and hemoglobin we've talked about, it transports oxygen, myoglobin transports oxygen in the muscle tissue, okay. What else? Membrane transporters, this is where we were discussing recently, talking about proteins that transport things across the membrane. Support, so collagen. Remember, collagen was what made up gelatin, okay? And that is the most abundant protein in the human body. It's basically, think of it as the glue in your body, okay? And it links everything together. Keratin, hair and fingernails, okay? I'm right here. Keratin, hair and fingernails, etc., etc. Motion, actin and myosin make up your skeletal muscles and your cardiac muscles and any other muscle in your body. Regulation, that's an interesting one. Your serum albumin. This is a protein in blood that holds water in the blood, okay, which is interesting, etc., etc. And storage, for example, ferritin. Ferritin is an example of a protein that binds iron and stores it in your body. So proteins are an extremely complicated class of molecules, probably the most complicated, and they do the most, the largest variety of things in the body. Okay, so again, here's our dehydration synthesis. If you remove water between two amino acids, you're going to form a dipeptide, just like a disaccharide, okay? One amino acid comes together with another one, and you get a dipeptide. And notice it happens between the acid group of one amino acid and the amine group of the other amino acid. And by the way, that's where they get their names from. The nitrogen is the amine side, the carbon with a double bond to an oxygen and an OH is an organic acid. So you put those two words together, amine and acid, you get amino acid. Okay, and the little funky dudes, let's go to green here, that make every amino acid different are these things called R, which we'll talk about in a minute here. Okay, so these are the common 20 amino acids that we have here, and they have names: serine, alanine, valine, asparagine. You might have heard of them before. For example, asparagine is processed by your body. It's very common in asparagus. There's a high concentration in asparagus. And that's what gives your urine a stinky odor after you eat asparagus. Very interesting. Primarily because of a lot of nitrogen that's in there. Okay. What other things might you have heard? Lysine, if you remember in Jurassic Park, if you ever saw that movie, they were trying to breed the dinosaurs lysine deficient so that if they ever got off the island, they would die. Okay. What others might you have heard of? There are many. Okay. But these amino acids, if you notice, they all have, let's go back to red here. They all have this acid group and this amine group, every single one of them. The only thing that's different is this side chain, or the R that we were talking about. It's different in each one of the amino acids, and that's what makes them different. Think of them as like train cars. Here's my train car, my box car. I've got a hook and a loop, okay, and the next car puts its loop around the hook, okay, and that's a loop and a hook, and that's how you join them up. So the two ends of the cars have to be different, but you can make what stacks up on the cars. I mean, the two ends have to be the same, but you can stack up whatever you want on each one of the cars. You can have grain cars, flat cars, box cars, whatever. That's kind of like amino acids. They're different, but the ends are the same so that they can connect to form the polymers, which we call proteins. All right. So when you're getting into proteins, the structure, there are several ways that proteins fold and have their different structures. You have hydrogen bonds. Okay, hydrogen bonds are simply this attraction between hydrogen and an electronegative atom like oxygen or something. And again, it's kind of like that Velcro idea. Well, it is the Velcro idea. Very weak in itself, but if you get lots and lots and lots of them, it can be very strong. You also have a disulfide bridge where any amino acids that have sulfur, like cysteine or something, can actually form a bond there. That's very, very strong. Okay, there's ionic bonds. All right, full charged ions coming close together, attracting kind of like magnets, stronger than hydrogen bonds. Van der Waals interactions, again, you don't have to worry so much about it, more Velcro-y kind of stuff. Because if I were to talk about what would be like Velcro, it would probably be like the van der Waals and the hydrogen bonds. They're not real bonds, they're more attractive forces. We won't worry about hydrophobic exclusion, okay? So these are the forces that cause proteins to twist and bend shape. So here are our structures. Primary structure we've talked about, and we usually abbreviate it this way. Primary structure is just the line of amino acids. Secondary structure. Here are two examples. You have the alpha helix and the beta pleated sheet. Different ways of folding and helicizing the amino acids, okay, in these structures within the protein. Tertiary structures, when you get all of these, notice here's an alpha helix here, and here's some beta pleated sheets here. When they're all coming together and twisting up in a big three-dimensional shape to be functional, that's your tertiary. And of course, quaternary is if you have more than one tertiary protein getting together to do a job. And the example was hemoglobin, which is a quaternary protein of 4 units that carries oxygen in the blood. Okay? And what do we have here? Okay, motifs. Just a real quick note on motifs and domains. Motifs, as it said in the book, scientists studied lots and lots and lots of different proteins, and they find a lot of proteins have very similar structures within them, even though they're different proteins. So a beta-alpha-beta motif versus a helix-turn-helix motif. You'll see these a lot in proteins because oftentimes there are different proteins that have to cross the membranes of a cell or whatever it may be. And these motifs appear again and again and again, and there's this genetic stability in them. And so oftentimes you can look at a protein with its motif, and you can find a lot of proteins that will comparatively have similar structures, all right? And you can use that to kind of look at the evolution of how proteins develop. That's really all you need to know about motifs, just common patterns. So think of motif as a pattern. Okay? And domains are simply areas of the protein. So if, for example, the best example we had of that so far is we're talking about, so if we have the membrane, okay, and we had a protein going through the membrane, okay, and this is our protein, you probably would have something like this domain here of the protein, and then these domains on the outside of the protein be more, so these domains, the red domains that I just drew, would probably be hydrophilic, the green would be hydrophobic because it's sitting in a membrane. So domains are just areas of the protein, okay? Okay, this was the cool one, chaperones. When proteins assume their primary shape and are sequenced into their polymers, they have to fold into their tertiary structure to become functional proteins, but sometimes they need help. And notice, we have our buddy ATP here. Okay, this doesn't happen by accident. Many proteins need to be assisted in the folding, and oddly enough, it's done by other proteins called chaperones. So just like chaperoning to a dance, the chaperones will help them, okay? And so they will bind to them and assist them in folding, or if they are misfolded, they can assist them in refolding to the right shape. That's all the chaperones are. Okay, and again, the idea of denaturation. We talked about this in the gelatin lab. If this is the properly folded protein, this is the denatured protein. Notice it's basically unraveled. Okay, it's come apart. All right, it's no longer possessed of these wonderful domains, motifs, beta pleated sheets, and alpha helices. It's just all come apart. It's a giant long chain, just like it's almost like just back to a primary structure. Whereas this is a nice tertiary structure. All right, so you've undone the tertiary structure. Even though you haven't chemically disassociated the amino acids, it's no longer a functional protein. It's a big long ribbon. This is in gelatin. You unravel the protein in gelatin, and then it binds with other proteins later on when you cool it down. Or when you cook an egg, it goes from tertiary and you unwind it, okay, the albumin in egg, and it turns into this big long ribbon, and then the big long ribbon binds with other big long ribbons when you're cooking it, and that's what causes the egg to gel up. All right. Okay, so lipids, the big ones here, we have the triglycerides molecule here. I'm going to reduce the size of this. Okay. You have glycerol up in the top, and you have 3 molecules of fatty acids attached to it. That makes a triglyceride, and they can either be saturated, like this one over here, or they can be unsaturated, like the one over here. And unsaturated simply means you have double bonds. Okay, and look what it does to the shape. Again, shape, really important. It causes it to go all crookedy. Okay, and that's really important in the body. Unsaturated versus saturated stacks really well, okay, and behaves, but there are some health consequences with that. Okay, other classes of lipids, terpenes, big long chains of carbons and hydrogens, all right, and you see these all over the body. Vitamins, vitamin D's, vitamin A's, retinol, things that help you see, you'll find that. But again, they're fats because it's mostly carbon and hydrogen, very little oxygen. And we probably recognize this, cholesterol. You see these 4 rings, okay, and that's your steroid ring structure. And then you can put all kinds of groups off of it. So anytime you see these 4 rings, a 6, 6, 6, and a 5-membered ring linked together, that's going to be a steroid. These are kind of like wax, okay? So these are very, very waxy, okay, in their structure. All right, these are your phospholipids. This is many ways to draw a phospholipid, okay? But it's composed of this molecule called choline, kind of like the word acetylcholine, phosphate, we know what phosphates are, glycerol that we just talked about, and 2 fatty acids. So it's kind of like you take a triglyceride, chop off 1 fatty acid, and then replace it with this phosphate and choline. What do you end up with is this part, this phosphate and choline part, is polar, and this fatty acid part is non-polar, which makes it really good at making these lipid bilayers. And again, of course, here's the final representation. You usually see it's a ball with 2 tails. Okay, very important. And they will spontaneously form membranes. So you throw this stuff in water, and it's a spontaneous process. They will organize themselves this way. And it is thought evolutionarily that this is where the first cells came into being, because these molecules would self-organize and form the membranes. Okay, so that takes you through a lot of the diagrams that are in the chapter, okay, and some of the very important vocabulary words that you're going to need to know. Okay, thank you.